Our research on ALS is focused on three aspects. One is we're very interested in looking at stem cells as a therapy in ALS and are trying to understand how stem cells can help patients with ALS. Secondly, we're very interested in how toxic exposures, for example, metals or pesticides that you're exposed to in the environment, can make a person more susceptible to ALS and the mechanism that underlies that. And finally, we're very interested in how what's known as RNA, which is a building block for proteins, changes in ALS. And these three areas are what's driving our very interesting and I believe novel research in ALS.
Epigenetics is the regulation of gene expression without the genetic information being altered. And epigenetic mechanisms are sensitive of the environment. So whatever is, if there's stress, it can be toxic chemicals, it can be weather, then these epigenetic changes will trigger changes in gene expression. If we can identify some of these environmental changes that trigger epigenetic mechanisms, then we might be able to modify these mechanisms in such a way that either we can change the onset of the disease in animal models, or we can maybe find treatment that might reverse those epigenetic changes, and hopefully we could find a treatment for ALS, or something that could actually give more hope to the ALS patients.
In my projects, I use samples from ALS patients and healthy controls to evaluate these epigenetic changes. I also study how other players can be involved in disease and affect epigenetic mechanisms leading to ALS. If we can better understand the causes of ALS, including epigenetic mechanisms, then we can pinpoint molecules that can serve as potential targets for therapy, as well as biomarkers for an earlier diagnosis, which is an unmet need in the field of ALS.
I studied amyotrophic lateral sclerosis, also known as ALS, particularly the role of the immune system in both driving and protecting against ALS. So the immune system is very complex. Not only is it used to defend the body against potential infections and pathogens, but it's also used in the repair response. So in the case of ALS, we're not really sure whether it's playing a protective or destructive role. Now, previous studies using immune suppression have actually made ALS worse. So we do know that there is an immune component and it may be playing a protective role. And so our goal is to use human patients and, based on their peripheral blood work, determine what cells may be playing a positive or negative role in disease progression.